Exome sequencing just looks at the exon portion of the gene. This is the part of the gene responsible for coding proteins. There are 180,000 exons, and they make up about 1% of the genome. As you can imagine, sequencing just this one part is much less expensive and time-consuming than doing the entire genome. Now, in order to look at just the exomes, target enrichment strategies must be used to capture the exome regions we want to look at.
There are a few different target enrichment strategies which can be used. The most common one, which we used in class, is the polymerase chain reaction. Nucleotides, template DNA, and PCR primers are added to the buffer solution. These primers tell the polymerase where to start and where to stop along the DNA strand. Then the polymerase is added and the tube is put into a thermocycler, which changes the temperature of the tube to facilitate the amplification of the DNA. The high temperature splits the DNA strand apart. Then the primers bind and the polymerase continues along, creating a complementary strand.
Another technique used is molecular inversion probes. They work by sending probes which are single-stranded DNA molecules with regions that are complementary to the section surrounding the exon we want to target. Once the probes attach to these regions, they will polymerize, which circularizes them. They are then released and part of the probe is cleaved, which inverts it. There are other types of target enrichment strategies like hybrid capture and in-solution capture, which work by hybridizing desired parts of the exons to identify and sort them and washing away the undesired parts of the gene.
After the desired segments of the genome are captured, they have to be sequenced. Some of the most common sequencing technologies include Sanger sequencing, which uses dideoxynucleotides that we learned about in class, the Roche 454 sequencer, the Illumina Genome Analyzer 2, and the Life Technologies Solid and Ion Torrent.
Exome sequencing has many useful applications. It's especially good for mapping rare variants that cause complex disorders as well as Mendelian disorders. A rare variant is a mutation that alters gene function, increasing the risk of severe genetic diseases. Because the mutation is typically removed by natural selection, the variants occur at low frequencies in populations. As a result, the exact location of the variant causing the disease is often unknown. Exome sequencing can be helpful in determining the cause of rare diseases because it is less costly and faster to analyze more data.
The very first rare Mendelian disorder whose cause was identified using exome sequencing was Miller's syndrome. It was first described in 1969 by a German ophthalmologist named Eckhard Jernet. The cause of Miller's syndrome was discovered by sequencing the exomes of 4 patients with this disease. Researchers predicted that the sequence would be previously undiscovered or unmapped, so they compared it to collections of existing SNP data. When they found no match, they could then identify it as a newly discovered mutation. They learned that the condition was occurring due to a recessive mutation in the DHODH gene, located on chromosome 16q22.
One of the most significant limitations of exome sequencing is that it only identifies diseases caused by mutations located in the coding regions of the gene. This means that only 1% of the entire genome is being sequenced. Although variants are more likely to exist within coding regions anyway, a lot of genetic data is being neglected, and non-exomic mutations could be going undetected. Despite this limitation, exome sequencing is far less costly and time-consuming than current techniques used for whole genome sequencing, and has very successful yield of variants for the tiny amount of genome being sequenced.